Hi, I'm Nikolai Nøjgaard, and I'm here to talk about graph transformation for enzymatic mechanisms. So, catalysis is a term describing a process in which the rate or the outcome of the reaction is influenced by the presence of a substance in the catalyst that is not consumed during the reaction. When we talk about catalysis, we talk about a chemical process that uses catalyst to enable some reaction. And this is important to emphasize because if we just look at the overall reaction of a catalytic reaction, it's not exactly clear how the catalyst is utilized in this process. To get a more complete understanding of a catalytic reaction, it's therefore normally more useful to depict the underlying process of the reaction, for example, by using a reaction network. In this process, it's clear that the catalyst is not some immutable object, but broken up only to then again later be reformed. For example, for the citric acid cycle that we have depicted here, we see that the catalyst, in this case the oxaloacetate molecule, kickstarts a sequence of enzymatic reactions that in the end generate a new oxaloacetate molecule. And we represent this by a reaction network containing a cycle that really underlines the catalytic nature of the process. More specifically for this talk, we'll focus on enzymatic mechanisms. So any reaction can be broken into its elementary steps that together constitute the overall reaction. And we call this sequence of steps the mechanism. So here we have elementary reaction 1, step 1, and elementary reaction 2, step 2. In enzymatic reactions, a protein or the enzyme functions as a catalyst for the reaction. Here, an enzyme is a chain of amino acids folded in 3D space. For the purpose of representing mechanisms, we really just ignore the structure and of the amino acid of the protein and only include relevant amino acids. For example, here we have included the tyrosine and serine for the mechanism that we have depicted. If we just focus on the reaction, it's not clear exactly how the enzyme plays a role in this process. However, when we focus on the mechanism, we directly observe how the amino acids are used and then later regenerated in the mechanism. This leads us to the problem that we will try and solve throughout this talk. Given an enzymatic reaction, how do we propose potential mechanisms for said reaction? Now, here we will, like in the previously depicted picture, treat an enzyme as a set of loosely connected amino acids making up the active sites of the enzyme. Now, the actual motivation of this is with the ultimate goal of enzyme design. Designing a full enzyme, however, requires us to actually have the structure which controls specificity and provides a stable active site that guarantees a proper causal ordering of the catalytic steps. While the network view does not really address this issue, it underscores, hopefully, during this talk that designing an enzyme also requires designing a multi-step catalytic process. So, given an enzymatic reaction, we do not a priori know the reaction network that represents that mechanism, and hence we need a way to compute it. For this, we built upon a graph transformation software called MIL to construct reaction networks. And to first understand what we do, I want to quickly introduce some core concepts in the framework. First, any molecule is modeled as a connected undirected label graph, and here the labels of the vertices are the atomic symbols, and the labels of the edges are simply the bond types. For example, here we have depicted graphs representing formaldehyde and glycolaldehyde respectively. Reaction rules are, on the other hand, represented by a double push-out rule. And these are graph transformation rules that specifies how we transform a molecule from one molecule into another molecule. And how it works is a bit technical and not too important for the overall understanding. But what's important is that we try to find an embedding of the left-hand side graph into some host molecule, and then we remove anything that is on the left-hand side and add anything that is on the right-hand side to construct a new molecule. And here, for example, we have the rules for ketonol isomerization and aldol addition, which is common in the Formose reaction, for example. These rules can continuously then be applied to molecules such as the formaldehyde and glycolaldehyde molecule that we saw before to construct a reaction network represented by a directed hypergraph. And here the vertices of the graph correspond to molecules and the hyperedges correspond to chemical reactions, or more formally rule applications that transform the sources into the targets. So we can in theory compute these reaction networks, but a key prerequisite to do this is that we have to have a collection of rules that faithfully represent the behavior we see in enzymatic mechanisms. Since enzymatic chemistry is rather unique, rules must therefore also be derived from this chemistry somehow. Luckily, there exists a database called the Mechanism and Catalytic Site Atlas, or just the MCSA for short, that does exactly this. The database contains a collection of enzymatic reactions along with a set of manually curated mechanisms for each reaction. And the aim of the MCSA is to provide a set of mechanisms that exactly models a lot of the behavior we see in enzymatic reactions. Uh, each mechanism is modeled as a sequence of electron pushing diagrams like the ones that we have depicted here that models a mechanism that you could see in the MCSA. And these diagrams contain a lot of information that we really were wondering if we could use to form the foundation of a rule set for modeling enzymatic chemistry. So really, if we just look at one of these steps or one of the elementary reactions, a electron pushing diagram already closely resembles a rule as we think of it in the graph transformation framework. To make this clear, we can abstract away the specific path the specific path of the electron, and simply focus on the charges and the charge changes of the bonds and atoms. And what we get is a rule like so, where the red edges are broken bonds, green edges are created bonds, and blue atoms and edges are bonds that change charge or valence. Now, this is basically already directly encodes a rule, however, not a very useful one, as molecules are already fully specified, and hence we won't be able to apply it to any other context. The core behavior of the rule that we know that we must encode, however, is the reaction center, meaning the atoms and bonds that change during the step. And we can use this as the basis of a rule. So this already fully encodes a rule that we could apply. However, if we just use the reaction center, we end up with rules that are very general, and therefore we will quickly end up in a bit of a combinatorial nightmare when we apply them, because we can apply them too many places. So instead, we augment each rule with a context that must be present before a rule can be executed, essentially embedding some knowledge of the chemical properties implicitly encoded in the environment around a reaction center. First, we assume that the active amino acids are crucial for driving a reaction step. If any part of the amino acid intersects with the reaction center, we include the whole amino acid in the context or in the rule. Next, we assume that the immediate surroundings of the reaction center are a significant driver of the reaction, and hence all atoms and bonds that are directly connected to the reaction center are also included into the rule. And finally, we compiled a set of chemical patterns that we deemed of chemical significance. And if any of these patterns intersected the reaction center, the whole pattern is also included. So for example, here the ester pattern is deemed an important pattern that we must include in our rule. Now, using this approach, we can construct a set of rules, and given some electron pushing diagram. And each step of a mechanism produces exactly one rule. And we can use these rules to represent each mechanism in the MCSA as a sequence of rule applications converting the reactions into the products. Here, each colored arrow is a rule and similar to the corresponding step, and each node is intermediate molecules. We can take each of such rules and put them in a set and declare that these rules can be used to model enzymatic mechanisms in general. If we were given the reactants of a reaction, we could construct a reaction network by simply repeatedly applying the rules to the reactants and its resulting derivatives to, for example, create a network like so. However, such a network generation strategy will quickly explode as we do not respect the stoichiometric information given by the actual reaction. What I mean by this is, imagine that M1 is a reactant and M2 is one of its derivative. Well, then we know that they will never be able to exist, these two molecules will never be able to exist at the same time, and therefore they will never be able to combine to create the orange rule that we see here that constructs the molecule M3. So this essentially leads us to essentially construct a state space graph on top of the reaction network. And here, what I mean by a state space graph is a graph where each node is a collection of molecules, i.e. a state, and each edge is a rule application describing how the collection of molecules in the state can change. As a concrete example, assume that we are given an overall reaction like so, and we represent reactants and products as a collection of molecules corresponding to a node in our state space graph. Now, from the reaction, we can see that we can ask what are the possible states we can create using the rules that we are given. And we might see that we can apply the green rule to construct a new state. And from this state, we can ask the same question and see that we can create two new states. And from these two new states, we can ask the same question and same question until we have a state space graph like so here. The hope is that at some point we'll find a state that can be converted into a product state. Indeed, any path from the reactant to the product corresponds to a mechanism which we can then propose. So this is, in theory, essentially a way to propose concrete mechanisms for a given enzymatic reaction. Now, we evaluated our method using the REA database that consists of 8,805 manually curated metabolic reactions, many of which are enzymatic, but with unknown precise mechanisms. Because the mechanisms are not given, we do not know the active site of the reaction, and hence we do not know what amino acids are present during the mechanism. To solve this, given a reaction, we created a set of 17 different amino acids plus their tautomers and created a state space by placing a single amino acid in the reactants and in the products. And we know that we can place a single amino acid in both because we know that the amino acid must be regenerated during the mechanism. We did this for each reaction and recorded if a mechanism could then be proposed. And indeed, for around 9% of the reactions in the REA database, we could propose a mechanism that used exactly one amino acid. Now, as a result, we get a table like this that shows the amount of reactions for which we could propose a mechanism that uses the amino acids. In this case, it's just 5 amino acids. And we, for example, see that for 624 reactions, we could use glutamate to propose a mechanism. And the bottom row shows the unique amino acids for said reaction. So what I mean by this is for 135 reactions, glutamate was the only amino acid we could use to propose a mechanism. What we see is that glutamate and histidine is really the most common amino acids that we could use, which is really not too surprising since these are also the most common amino acids that we find in the MCSA. So due to the number of experiments we had to run, it's not currently feasible to try and see if we can propose mechanisms for all combinations of amino acids. But we can, however, query for mechanisms that exhibit a specified complex behavioral motif that we find in the MCSA by carefully selecting the set of catalytic amino acids that is present when we expand the state space. So one such behavioral motif that we can examine is that we find in the MCSA is the joint action between histidine and serine. And just in short, what we see very often in the MCSA is that the histidine deprotonates the serine, which allows the serine to attack the substrate, attaching itself and forming a complex. Now, we can query for such behavior by simply just adding histidine and serine to the reactants and products, and then just simply see if we can expand a state space and find a mechanism. And we do indeed see, for example, for the reaction that we have here, which is in the REA database, that we get a reaction exactly like this one, that exhibit exactly the same behavior as the one I just described. So in conclusion, we can construct rules from a given set of mechanisms that are represented by electron pushing diagrams. We can use these rules to construct state spaces, and we can use these state spaces to infer mechanisms of a given enzymatic reaction, at least to a limited extent. This should be emphasized that this is the very first steps. There exist a lot of other things that we can do to work on, like for example, the rules right as they are right now are fairly crude, and we could put a lot of work into refinement of these rules. And we also need to add a lot more complex chemistry as cofactors and more amino acid combinations. But this really needs us to develop better algorithms. But again, this should be emphasized that this is really a proof of concept about the the applicability of our approach. So if any of this sounds interesting, my colleagues, specifically Daniel Merkel, have gotten accepted for a Novo grant spanning 6 years, focusing on the design of microbiomes that uses a lot of similar techniques as discussed here. And they are currently looking for a lot of postdocs. If anyone is interested or know anyone that is interested, please mail Daniel Merkel as the email suggests here in the bottom left. Right, that's my talk. Thank you very much.